Hello, class. So today we're going to be looking at variations in spider offspring models. And the first thing that you always need to do is follow the instructions. So it says here, carefully review the proteins each spider has. And you might be thinking, proteins? What are proteins? And the proteins are located here inside the cell, and they vary in shapes, or as we call gene versions. So decide which gene version they have. And we're also going to add annotations, notes, or label anything that we'd like here in this model, and hopefully use these science words: gene versions, proteins, those scientific terms, homozygous and heterozygous, as well as traits. So let's get to it here. And it might be a good idea for you to zoom in. So there's a zoom in here. It's under the View tab. It looks like a magnifying glass. So you click there at 100%, and you might even want to zoom it in a bit more. There we go. So as we stated before, these are the proteins, and they look the same. Right? They all look the same. So those are the protein molecules, and if we look down here, which one does it most closely represent? Well, I know you can't see the other two, but it closely represents gene version F1. So we're going to type here F1. And let's highlight it, use this bucket, and oops, transparent. Let's highlight it in red. There we go. And there's only one gene version. It's the same gene version, F1. So we're going to put F1 for both copies. So we're going to click on text box. And we have to make the box smaller. And type F1. Make it a little bit larger so it can fit in. There we go. Click on this text color. Make it that nice red so it will stand out. And that was for Offspring A, and Offspring A had low silk flexibility. And now we're going to be looking at Offspring B, which has a medium silk flexibility. And Offspring B, let's lower the screen a little bit more so you can see the different gene versions that I was talking about. So again, we're looking at proteins, and we're looking at these 1, 2, 3, 4, 5 protein molecules. There it is again, that same protein molecule that was in Offspring A, right, which is gene version F1, right? It's this one here. Mr. Lopez tried to draw that for you because it was not, it wasn't added. So F1. Actually, I need to type that in, but it's going to be F1. And we have another protein molecule here. right? We have two different protein molecules. They are different. They are not the same. And this one looks to be gene version F2. So we're going to type F2 here. So F1 and F2. We're going to highlight. We're going to use text color and do the same thing over here on this side. Click on text box. I'm going to make the box smaller. I have to make the box smaller or else it will type into the other into this other area. So it's very important that you do that. So this is F2, F version, gene version F2. And if you look at these two offsprings, these gene versions are the same. And that's what we refer to as homozygous. And now what we're doing is we are annotating. Let's put this in a nice blue and bold. It's a very important word. And these gene versions they're different, right? We have F1 and F2. There are different protein molecules floating around this millions of cells that are in this spider offspring B, which has a medium silk flexibility. And what do we refer that as? That's right, heterozygous. So let's type heterozygous here. Let's minimize the box. And also highlight, use text color, click on B, which means bold. And let's move on to the third offspring, which has low silk flexibility. Now remember, this offspring has low silk flexibility, and this offspring C, offspring A, also has low silk flexibility. They both have low silk flexibility. Do you think that they will carry the same gene versions? We're going to find out. Let's look at the gene copies it carries. Here they are. If we look, we see the same protein molecule as offspring A, right? Same gene versions. So, which is F1, right? So we're going to clear this and we're going to type in we're going to look for the text box and we're going to type the same gene versions here for offspring C. F1. And do you have to change the color? You don't have to change the color. It's quite all right. As long as you are filling these in, that's all that really matters for you to get credit. We're going to click on text box, minimize the box, and we have F1. We're going to highlight, click on text color, and we now have one last Offspring, which has high silk flexibility. High silk flexibility is this spider is much different than the others because it has the highest silk flexibility. And do you think it will have different protein molecules than an Offspring with low silk flexibility? Well, let's find out. So if we look, okay, so if we look here, we see it is this protein molecule is F3, and this protein molecule here is F2. Sorry, I can't point it. I don't know where my pointer went, but that's what we have. We have F1. Sorry, F2. And going to click on text box, minimize the box. I know this can be a little bit annoying, but there's no way around it. And this is going to be protein version F3. We're going to highlight, click text color, highlight, click text color, and of course this is being recorded, so you can always rewind as many times as you need to. And if I zoom out, I have completed the lesson. I am done. Yay. And this is all that needs to be turned in. So if you have any questions, feel free to message me, and have a great day.